Hello everyone, my name is Shubham. My data science project topic is bacterial species identification using k-mesh with machine learning. The main purpose of this project is to identify the bacterial species using genomic data or DNA sample of bacteria. Fast identification of bacterial species is necessity when the bacteria is pathogenic, and delay in identification of such a bacteria can cause serious consequences on patient's health. The advantage of this project is it identifies the bacterial type in less amount of time, which helps to treat patient. The data that we have used for this project is taken from Kaggle site. The actual dataset is simulated data of DNA k-mesh. For classification purpose, we have used different classifiers like logistic regression, naive Bayes, KNN classifier, and random forest. Why bacterial species identification? A lot of bacteria are considered as a bad for humans and blamed for most of the global illnesses and fatalities. Identification of such a pathogenic bacteria are helpful for diagnostic procedure. For example, identification of bacterial species is very important for the septic shock patient since the mortality rate rises by 7.6% per hour. Traditional way of classifying bacteria. Traditional approach for bacterial classification is based on physical characteristics of bacteria. Drawbacks of conventional method. Sometimes same bacteria used to exhibit different physical structure which leads to wrong identification, and this traditional method is time consuming and very expensive. Solution to the traditional method. Instead of identifying bacteria based on their physical characteristics, they must be identified by using genomic sequence. So the chances of wrong bacterial identification is very low irrespective of the physical structure of bacteria. Methodologies. By using machine learning algorithms, especially classification algorithm, the bacterial species can be identified quickly within higher accuracy. Advantages: fast identification, higher accuracy, cheaper compared to the old way of bacterial identification. The actual data for this project, I got it from the Kaggle website, and the methodology which used to generate this data through simulation is explained in this particular paper. Data generation, as per the paper that mentioned in the last slide, the basically DNA sample is taken, and that DNA sample is converted into 10 mers, and each of the 10 mer is each of the 10 mers block optical content is read, and then that block optical content is converted into the bins according to its fractional base composition. Similarly, a randomly generated fractional base composition spectrum is subtracted from our main fractional base composition spectrum, and which generates the spectrum of deviation. This is the actual dataset that I have downloaded from the Kaggle website. It has 286 features and one target variable of categorical type, which represent 10 different species of bacteria. It has 200,000 records, and each feature represents the histogram, and each data point represents the relative frequency of the histogram. As I said in the last slide, that it's a spectrum, therefore data point values are float. As a part of EDA, I checked the frequency of each class in the dataset, and I found that records for each class exist almost equal times as other bacterial species. The next step that I have performed is data preprocessing, in which I have removed all the duplicates records from the dataset. So there were 200,000 records in my dataset, out of which 76,007 records were duplicate. So I have removed all of the duplicates record in order to avoid skewed performance of the model. And the next thing that I have done is data encoding on target variable. Since my target variable was a string, I have converted into the numerical values. After data preprocessing, I have divided the dataset into training dataset and testing dataset, 75% and 25% respectively. Then for each of the machine learning model, I have performed the hyperparameter tuning. So for that, I have defined the range of values for each of the hyperparameter. Then I have choose the cross validation method, which was stratified cross validation. And after performing the hyperparameter tuning using grid search CV, I found the best parameters for each of the classification model. Then using those best parameters, I have trained the whole training data, and I have predicted the bacterial species on this testing dataset. I have performed the hyperparameter tuning in order to find out the best parameter for the classification model in order to increase the accuracy while classifying the bacterial species. So for logistic regression, I found C is equal to 1000, penalty L1, solver linear as the best parameters. For random forest, I found max depth, means the depth of the tree is 16, number of trees 256 as a best parameter. But in case of naive Bayes classification algorithm, I didn't perform the hyperparameter because there is no hyperparameter in naive Bayes. For KNN classifier, I got number of neighbor as a 9, metric Minkowski, p-value 1, and weights as a distance as a best parameter for the KNN classifier. After predicting the bacterial species on testing data, we can see logistic regression gave 89.75% of accuracy, random forest gave 96.89% of accuracy, naive Bayes gave 50.19% of accuracy, KNN classifier gave 95.99% of accuracy. Through confusion matrix, we can see how many times model predicted the right bacterial species and also the count for each misidentification with other bacterial species. According to result, the best model is random forest for predicting the bacterial species. During this project, I have used various tools and technologies. I have used Python programming language, pandas and numpy libraries, and for visualization purpose I have used Seaborn and Matplotlib libraries. As a integrated development environment, I have used Jupyter Notebook. The first conclusion is the classification using bacteria's genomic sequence is more effective compared to classification using bacteria's physical trait data. Second conclusion is classifying the bacteria gets simple as the length of DNA sequence grows. And the third conclusion is random forest algorithm is better at classifying the bacteria compared to all other classifiers like KNN classifier, logistic regression, and naive Bayes. This project can be further improved to identify the antibiotic resistant gene present inside of bacteria by comparing the DNA sequences to available bacterial DNA sequence using machine learning algorithm.